Welcome to the PPH Plus webinars. My name is Dr. María Proupín-Pérez. I am project leader at PPH Plus, and we are going to talk today about quality by design and the risk-based approach in clinical trials. In this webinar, we will start presenting the problem that clinical trials are facing today concerning quality. We will check the solutions that regulatory authorities are promoting worldwide, and these are mainly the quality by design and risk-based approach. And finally, we will take some conclusions with the intention of learning how to do things better in the future. We need to start asking ourselves, what is quality? And quality is nothing else but fitness for purpose. So you need to define the purpose in such a way that you make fitness for purpose feasible. Let's try to avoid complexity from clinical study designs. When we avoid complexity, we make fitness for purpose feasible. So what is the situation today? I've depicted the situation today in this cartoon. We have a few guys falling down the cliff, and one of them is asking questions, and other is responding, "Don't waste time with insubordinate questions." We do not waste time. We don't want to waste time in the beginning. We find this time, this planning time period, not useful. We think it is wasting time, and that's the main mistake. Neglected quality is not possible to be changed or to repair it or correct it afterwards. Keep in mind that quality cannot be built retroactively. Once these guys fall down the cliff, you cannot bring them back to life. I want to support this argument with facts. So I'm showing here in this slide the results of the GCP Inspectors Working Group. These are inspections conducted in year 2015. There are 75 inspections conducted worldwide at 9 sponsor sites, 57 investigative sites, and 9 other sites. And you find here on the right side that in total they reported 555 major and critical findings. And that is only in 75 inspections. And if you pick up the 3 main categories concerning trial management, general issues, and sites, you will find that concerning sponsor performance directly, trial management, they have reported 121 findings. So this is non-compliance. On top of that, and continuing with inefficient designs and quality management that is inappropriate, we find out the following issues. 50% of the sites do not reach the planned recruitment targets. So half of the sites are not reaching your planned recruitment targets. More than 95% of the clinical trials do not end on time and on budget, and that is mostly all your trials that are ending late. And then 90% of the studies meet recruitment goals, but at the expense of mostly twice as much time as originally planned. And these delays are producing gigantic costs. We have too many avoidable protocol amendments, with the first amendment even before enrolling the very first patient, and that is because we are rushing towards the first patient in. As shown in my cartoon, we don't want to waste time in the planning phase asking insubordinate questions. We don't want to ask questions in first place, and that's the big mistake we are doing nowadays. Additionally, we have compliance flaws as shown in the report from the ICH Inspectors Working Group. These compliance flaws are causing delays and additional costs. All of this together is telling us we need to change something. We need to establish a new objective, and the new objective is to get it right the first time. In order to achieve this, we need to minimize the need of rescue measures. We need to minimize rework that is laborious. This means that we need to seek prevention over correction, and we need to focus on what really matters. And per definition, as requested by the regulatory authorities, the FDA and the European Medicines Agency, the EMA, we need to focus first on patient safety and data integrity. So this is changing the currently approach drastically. How can we improve then clinical trials designs? We need to introduce quality by design. We need to build quality directly into the clinical study protocol. The draft addendum of the ICH GCP guideline is requesting quality management to include the efficient design of the clinical trial protocol. This is also involving data collection tools and procedures, as well as the collection of information that is essential to make decisions. This is now requested by regulatory authorities again. And how can you implement quality by design? You need to start designing your working space. The concept of design space has been implemented in pharmaceutical companies for many years. It is mentioned and described in the ICH guideline on pharmaceutical development, Q8. And by definition, it is the common region of successful operating ranges. How can we understand this? Let's take the enrollment or the inclusion criteria as an example. Let's say that in this graph on the left, you have represented your inclusion criteria. This white area represents what patients you can include in your trial, and the gray area represents what patients should be excluded from your trial. According to your inclusion criteria, you can enter all these patients. But then on top of that, you include this parameter 2, which can be the exclusion criteria, which is describing this extra area on the right. And when you overlap the inclusion and exclusion criteria, you end up with this smaller area, which is your design space. You should operate within these limits defined by these overlap areas. So when you are very restrictive, you have very restrictive inclusion criteria, you make this area much smaller. And if you add on top very restrictive exclusion criteria, then you are making this area even smaller. You have to try to make your inclusion-exclusion criteria as flexible or less restrictive, so that you are making your operating range, this region for successful operating ranges, bigger. That makes your life easier, and it increases your chances of succeeding with your trial. What has this to do with the risk-based quality management? Well, the risk-based quality management is nothing else but asking you to evaluate the risk of not falling inside or not operating inside the design space that you have designed yourself. According to the draft addendum to the ICH GCP guideline, The methods used to assure and control the quality of the trial should be proportional to the risks inherent in the trial and the importance of the information collected. This means that your quality management system should be adjusted to the probability of operating outside your design space or the risk of operating outside your design space. So please, let's design this space in such a way that fitness for purpose is feasible. When you design this space, you are designing your purpose. Let's do fitness for purpose feasible by decreasing the risk of falling outside your design space, of operating outside the successful operating ranges that you define yourself. And what is the main advantage of this quality by design and risk-based approach? The main advantage is that they provide continuous improvement. They follow the plan, do, check cycle methodology. So you need to start planning, you need to say what you do, then you need to do what you say. This means you have to follow the plan, then you have to prove you're following the plan. You need to be able to show that you have been compliant. And if something goes wrong, you have to act timely, you have to implement your mitigation actions, you have to keep things under control, and if things are not under control, you need to plan better again. This is the continuous improvement that this quality by design risk-based approach is facilitated. So you plan again, so you redefine your design space to make fitness for purpose feasible. And then you start again, you start do, continue with the plan, do, check, act methodology all over study conduct. But be careful, because it's not enough that you implement the plan and that you correct things. You need to correct actions, correct processes and procedures timely. You have to improve processes timely. That is one of the inspections findings of the GCP inspectors working group in year 2015. As an example, late protocol amendments are not satisfying inspectors because they are not done timely. So it's not enough that you are compliant or that you improve the system, you have to do both things timely. What is the risk-based quality management system? What is it entailing? I'm showing you here the system overview provided by the ICH GCP addendum, the draft version. We need to start first with the identification of clinical processes and clinical data. What is a clinical process? A clinical process is any process that is related to patient safety or to, for example, your endpoints. What are critical risks? In a second step, you will need to identify the risks that are threatening your critical processes and critical data. So you identify first what is critical in your clinical study, and then in a second step, you identify the risks threatening those critical processes and critical data. In the third step, once you have identified the clinical study risks, you proceed with risk evaluation. And risk evaluation is important because it prioritizes risks. You use this risk evaluation to check what risks are more relevant for your clinical study. And why are you doing this? You're doing this in order to plan your quality and risk management system in such a way that you are intensifying activities that mitigate or avoid the most critical risks. So once you know the priorities, you can adjust your monitoring strategies, your quality and risk management strategy, so that these relevant risks are mitigated or avoided. So you have to proceed with the preparation of a monitoring plan that is intensifying activities on those risks that are more relevant for your clinical trial. Once you have this integrated quality and risk management plan and the monitoring plan in place, you can move to the clinical study conduct phase. And during clinical study conduct, you will proceed with risk control. So you need to implement the plans. You need to follow the monitoring strategy. You have to proceed with quality assurance too, to make sure that risks are kept under control. The fifth process defined by the ICH GCP addendum is risk communication. Risk communication is related with the alerts, with the way that you utilize to communicate partners that they need to take action concerning risks. So once you have defined your monitoring strategy, you have to include the information concerning when alerts and to whom alerts should be sent, so that mitigation activities are taken in time. Again, you have to do things timely. It's not enough that you do it, you have to do it timely. You have to seek prevention over correction. The next process is risk review. Risk review is required for every single trial. Risks are changing in real life. There is no single trial where no new risk will appear. So in all single trial you will find new risks during study conduct, and for this reason you need to always proceed with risk reviews, frequent risk reviews that enable you to identify new risk or to change the risk priorities according with the experience gained during the trial. This is directly related with continuous improvement of your quality management system and your monitoring strategy too. The last process is risk reporting. You need to proceed with risk reporting not only during study conduct. In the end of your clinical trial, you will need to show how compliant you have been with the plans that you have defined in this planning phase, and show what deviations from this plan you have observed and what you have done concerning these deviations. So again, you have to show that you define the purpose correctly and that you have achieved fitness to purpose. Let's focus now in these first two processes, the identification of critical processes and data and the identification of critical risks. I'm showing here a tool that is available online for free. This is the Quality by Design Critical to Quality Factors Principles document created by the Clinical Trials Transformation Initiatives, the CTTI. The CTTI Quality by Design Project Critical to Quality Factors Principle document is a brainstorming tool. It is not a checklist. They say that in every single page of the document. This tool is enabling your teams to come together and discuss or design factors so that they think and they take time to check if these design factors are proportional to the objectives that your trial has established. And as an example, I took the factor data quantity. How much data you need to collect. Some of the questions this factors principle document is asking are the following. What is the justification for data collection if not classifiable? For example, if you cannot classify data as endpoints or safety, do you really need this data at all? So you may be able to withdraw data from your protocol so that you do data collection easier and you make the life of your investigators easier. Another question concerning data quantity is, could any exploratory endpoints be eliminated to simplify data collection? Again, You have exploratory endpoints. Are you ever going to analyze them? If you cannot take any conclusion from these exploratory endpoints, do you really require to collect this data? Again, try to make the life of your investigator easier so that you increase compliance, because they will have more time to do things better. They may provide better quality concerning critical data that is for you more relevant than these exploratory endpoints. Another example, for which data points can a greater error rate be tolerated, and for which is there a more limited margin for error? You need to establish your tolerance limits. You need to establish under and out of control limits, so that you are aware of how relevant an error is for each kind of data or for each type of data. For example, for you it is more relevant to have accurate data concerning endpoints than, for example, concomitant medication in a trial where concomitant medication is not entailing a risk for the patient's health. Another example, have site personnel given feedback on CRFs and proposed timing of assessments? Have you checked if the timing of assessments has been followed in past trials, for example? If not, if you see that the time frame established for visits and for the assessments is not respected, it might be a compliance burden in this new trial. So try to adjust this timing of assessment in such a way that you make fitness for purpose visible. You can implement a wider time frame for assessments so that it is more likely that investigators are compliant. This is quality by design. How are pharma companies dealing with this risk-based approach in clinical trials? I'm picking up the example of a big pharma company in oncology trials. They have implemented this risk-based approach in quality management in all their phase 1 to 3 clinical studies. They started in year 2015. What they did first was to create standard operating procedures, or SOPs, on management of clinical study risks to provide guidance on how to proceed with risk management. So they started with process maps for risk management. So they described the process of risk identification, assessment, control, review, communication, and report. They define functions, roles, and responsibilities. They define who will be responsible for risk, who will be accountable, who needs to be consulted, and who needs to be informed. So they assign a risk owner for each risk or for groups of risks. For example, it could be the project manager, it could be the medical advisor, it could be the data manager. Depending on the type of data or the process, you will assign a risk owner that should be qualified to deal with these specific risks. And you will have contributors, you will have people that will be helping this risk owner to resolve risks. They will be responsible, they can be consulted, and they may need to be informed in order to implement the risk management strategy successfully. Finally, and this is very important, you will make use of knowledge. You will have reviewers of the clinical study risk management plan. These senior functions will be able to support you and to help you to improve the plan from the start. You have somebody to consult with that provides experience, and that's very valuable in this risk-based approach, quality management. And finally, they provided a risk management plan template. So they have a clinical study risk management plan template enabling everybody to start working with, so they ensure they have a kind of solid guidance to meet some kind of standards. How can you implement all of these? How can you start, for example, assigning risk owners, etc.? You should check your risks. You should see how you organize your strategy, how you define your strategy. I'm showing you how it is widely done following the initiative of Transcelerate Biopharma. This is a non-profit organization. They have been pioneer providing training materials and free materials that are very helpful concerning the implementation of the risk-based approach in clinical trials. And I'm following here, showing you here more or less their approach, their strategy. What they did, they divided risks, or they group risks in risk areas or categories. And in this case, I grouped them in 3 big blocks or areas. You can group them as you wish. The important thing is that you go through all these factors, design factors. So you have first place study design factors. These are directly related with the clinical study design. Then second block is related with the operational aspects. These are your framework. This is your framework where you need to operate that your company and your location is offering to you. For example, concerning regulations, standard operating procedures of your company, the technology you have available, etc., etc. And third block is concerning the inherent characteristics of your investigational medicinal product. So, for example, under the block of study design aspects, you will find the comparator. You will need to ask yourself, if you are using a comparator in different countries, first of all, is this comparator approved or authorized in all the regions? If not, can you find another comparator that is authorized in that specific region? If not, can you try to find another comparator, or just change the region, find another region where your comparator is authorized? Another example, blinding procedures. Are you planning to proceed with interim analysis of data? If yes, how are you going to prevent unblinding during this interim analysis? And concerning operational aspects, you will need to check, for example, what technology you have available. Centralized monitoring is of great value in this risk-based approach because it enables the access to data from remote locations, and this is helping you to detect pitfalls before they really materialize. So you can proceed with preventive actions much easily if you have a technology in place that enables you to get access from data remotely. But now you have to keep in mind that the new addendum to the ICH GCP guidelines requesting you to have SOPs concerning the validation of IT systems. So it's not enough that you have the technology in place, you need to check what other demands you are facing concerning the use of technologies. Well, these are just a few examples of how you can start identifying risks. Once you have identified risks, you will proceed with the risk evaluation. And this is the third process mentioned in the ICH GCP addendum. This risk evaluation is quite tricky. You need to be able to do it systematically, and you need to obtain risk priorities as a result of this evaluation. I would recommend you to follow the Translate Biopharma Inc. recommendations. These are for free available. They are freely available online. As I said, this is a non-profit organization that is comprising the biggest pharma companies. So it is recommendable to consult this document. It may be the case that this document will be used, these documents or Translate Biopharma tools will be used as an standard in the future for the implementation of the risk-based approach in quality management. Why are we doing this? Why are we proceeding with this risk assessment and prioritization? We are proceeding with this risk assessment to avoid being a candy like this one represented on the left. We have two candies in front of a school and there is a storm. So the candies are on the road, they're getting wet, and they are, apart of getting wet, they are sitting in front of a school. In this school, there are thousands of kids that are about to go out and approach these candies. So the main priority here for these candies, or the main risk for these candies, is that kids will find them and eat them. They will be killed. They have no preventive measure that will prevent kids to eat them. And the candy on the left is worrying about the rain. The rain getting wet is a secondary risk. The rain may spoil the quality of the candy, but it will not kill it. So we are doing this risk assessment and prioritization process because we want to be able to do these two things. The first thing is to establish the key priorities. In this scenario, the kids that are about to run over the candies. The first risk is, the major risk is to get killed by the kids, so that should be prioritized. And the second reason why we do this is to think and to find the best or more appropriate mitigation actions, risk mitigation actions. So in this case, the wrapper is waterproof, but it doesn't prevent the kids to open the wrapper and to eat the candy. So in this scenario, the preventive action that will be really effective will be in first place that the candies are not in front of the school, or that, for example, they are locked into a kind of bottle that the kids are not able to open. That's the reason why we do a risk assessment and prioritization step. We need to first find what risks are relevant, more relevant, and secondly, what preventive actions are more effective. Let's take an example of how to implement risk management in clinical trials. I picked a study on psoriatic arthritis. We use a risk assessment and categorization tool slightly modified. It is the RACT Plus. It is based on the Translate RACT, but we have modified it slightly. This RACT Plus has risk classified in 13 categories and 66 questions in total. In this example, I pick up the risk of a patient suffering a serious side effect. So for each risk, we check the impact of the risk, that is, what will happen if a risk materializes. We put ourselves always in the worst scenario. The second attribute we check is the probability of a risk to materialize. We check how likely a risk may occur. And the third attribute is the risk detectability. Is how easily we can observe if a risk has materialized, and also we check if it is possible to react before having an impact, for example, on patient safety. Concerning the impact in this specific example, we observe that patients may suffer an anaphylactic shock. So the side effect, the worst side effect, is an anaphylactic shock. This is a high impact as patients may die. So we move to probability. Concerning probability, we did some research and we found out that the occurrence of anaphylactic shocks is between 1 and 0.01% of the subjects. So based on this frequency of occurrence, we assign a probability score of 2. I should have said we used a scoring system from 1 to 3, 1 being the lowest risk, 3 being the highest risk. So in this case, we assign a medium probability score. Then for detectability, we provide another score, and in this case, detectability should be checked carefully. We have seen that the patients are treated in a controlled environment. This means that you can detect easily if side effects occurs or not. However, an anaphylactic shock may develop very quickly, and therefore you need to react very, very quickly too. And since the warning signs are before serious impact on health of subject, but you need to react quickly, we provided a medium detectability score. So it meets medium detectability criteria. And what we do next is to multiply all these scores to obtain a final score of 12. This is, for us, the score of 12 is meeting the medium risk criteria. And what does this mean? We have established in first place a system to evaluate risk priorities. That is an ICH demand now. And secondly, we have checked what risk level is to be kept in mind concerning side effects. So you need to plan activities now that are adjusted to this total score defined in this risk assessment and categorization. Once we have done this, you will have as key outcomes a risk register, that is a record of prioritized risks after identification of critical and non-critical study data and processes, that is with risk tracking during study conduct. So you have this risk register and you know what risks are more relevant for you, and you will use this risk to track them during study conduct. Then you have an integrated quality and risk management plan, and you have your clinical monitoring plan. These two plans have been adjusted to the risk priorities you have established. And finally, you have your clinical study protocol. And watch out, your clinical study protocol should be developed in parallel with this integrated quality and risk management plan and monitoring plan. You need to make sure that you implement quality and mitigate risks already during the study protocol preparation. So you build quality in the study protocol in first place, mitigating avoidable risks. And all this process from draft protocol to the obtain of all these key outcomes may take from 3 to 4 months according to the feedback received from big pharma. And now, once we have all these plans in place, we can proceed with the study conduct. And during a study conduct, again, we need to implement the plans. We need to be compliant. And I want to remark again that the draft addendum to the ICH GCP guideline is requesting the sponsors to develop a systematic, prioritized, risk-based approach to monitoring clinical trials. And that's the reason why I've shown you all these tables before. You need to proceed with a systematic approach in first place to identify and prioritize risks in order to plan your monitoring approach. And secondly, I would like to remark that monitoring is not enough. You need to have risk control and quality assurance systems in place, systems that will enable you to control that the written SOPs are being followed. You need to check for each non-compliance detected during study conduct that you are implementing the corrective actions that are appropriate. You need to check for a root cause. Now the ICH GCP addendum is literally requesting you to proceed with a root cause analysis for each detected case of non-compliance. So you need to proceed with a root cause analysis and implement CAPAs, corrective and preventive actions, timely. Now the ICH GCP addendum is requesting sponsors to have partner agreements in writing. This is not new. There are many things that have been done during the last years, but we need to keep in mind that the ICH GCP was created in 1996, that is 20 years ago. The new addendum is only reflecting what has been changing in the last 20 years. So what can we say concerning the risk-based monitoring plan? What components you should have there that will enable you to implement the most appropriate monitoring strategy? Your plan should be clear concerning the specific monitoring activities. Centralized on-site monitoring and source data verification should be clearly defined. You should provide guidance on what tools, logs, or templates should be used, as well as how events or results of the monitoring, how these events or results will trigger changes in the plan monitoring activities. So you can proceed with continuous improvement of your monitoring strategy. Again, this monitoring approach is adaptive. It is changing according to experience. Then you should keep in mind you have to define risk indicators. You should have defined risk indicators in such a way that you are able to know how to identify critical deviations or failures. Again, you need to define tolerance limits in your clinical monitoring plan so that your teams are able to check if processes and data are within the acceptable ranges. If you have critical data and processes outside the acceptance range, then you had to define, if possible already in the monitoring plan, your preventive actions or your corrective actions. You should have the review procedures in place so that you can change your plan, making good use of emerging knowledge and experience gained during study conduct. Concerning the tolerance limits, the ICH GCP addendum is stating the detection of deviations from the predefined quality tolerance limits should trigger an evaluation to determine if action is needed. What does this mean? I'm showing you here a control chart. You have to be able to measure risks. As shown in this graph, you have to be able to show numbers. You have to provide predefined quality tolerance limits. In this graph, you see here the upper control limit and the lower control limit on the bottom. Everything that is within these two limits is under control. Anything that is outside these limits is out of control. So what does this mean? It means you are being compliant if you keep values within the limits. You are being non-compliant if you are outside these values. Again, you are defining compliance yourself. You are using this concept of design space. If you are generous with these upper and lower control limits, then your chances of being compliant are increasing. And a third aspect is that you have to be able to trigger an evaluation. You have to be able to communicate when something has to be, or action has to be taken, or something has to be done in order to prevent or correct some issues. This communication is represented in this graph, depicted in the graph as upper warning limits and lower warning limits. So you see here, when these lower warning limits are reached, your team should be informed. You can have an automated system that sends emails. It might be a person that is responsible for checking if these limits are reached or not. You should establish the right approach, and this will be effective. You should be able to provide this information, provide these alerts timely enough in order to implement actions effectively. And again, you define the strategy. You can define these limits and how you communicate risk differently. For example, you can use the 7 rule, the 7 warning rule. It means that you check if the variability is not the expected. So you have, for example, a set of 7 data that are on the lower side of your graph. It means that there is a kind of systematic error. So you define your warning, your communication system, your how you want to trigger alerts, and you define these quality tolerance limits. This is very important. In the end of your trial, you have to be aware of this. In the end of your trial, you will need to report the quality management approach that you have implemented and how successful you have been with the implementation of this approach. So you need to summarize all important deviations from the predefined quality tolerance limits. So you have to design your space, as shown in this graph on the left, in such a way that these control limits and the control limits, you have to show how flexible you are and try to do it as simple and as feasible as possible. You define your purpose in such a way that you make fitness to purpose feasible. Again, you have to check what quality tolerance limits you are establishing so that you make compliance feasible or unfeasible from the start. Saying this, I'm now reaching the conclusions. The main messages I want to transfer you with this presentation are, in first place, quality and risk mitigation has to be built directly into the protocol during the study planning phase. You have to implement quality by design. You define the purpose so that you make fitness for purpose feasible from the start. Second place, be aware that the methods used to assure and control the quality of your clinical trial data should be proportionate to the risks inherent in the trial. This is requested now by all regulatory authorities. Make sure that you are able to measure the risks you have identified. These risk metrics are called risk indicators usually. Take advantage of the continuous improvement of the quality risk management system. You should improve it continuously. Make use of this knowledge gained before, during, and after the implementation of the quality risk management system into a clinical trial. Make use of the data you know. You cannot prevent all risks. That will be impossible. That's an utopia. But you can prevent those risks that you already know. And that's the objective of the risk-based approach. The cost of quality. You may think the cost of quality are going to increase enormously. Be aware that they will be compensated by enhanced compliance. And again, quality cannot be built retroactively. There are many things that you can't correct by rework. that's not working always. And secondly, you will optimize the study outcomes. There is a very famous phrase saying the quality will be remembered long after the prize has been forgotten. So keep that in mind. Taking the plunge into the risk-based approach requires some effort, but it will alleviate the need for hasty and unsatisfactory study rescue measures. It will prevent you to need rework. Once I said this, I'm providing you with a kind of toolbox with clinical study protocol templates that will, in first place, help you to build a protocol, making sure that all key factors are included so that you have a structure that you can use. You don't need to reinvent it from zero. You will be able to take advantage of protocol templates provided by the FDA, by the UK National Health Services clinical protocol templates. Swiss Ethics has its own protocol synopsis template. You have as well risk assessment tools freely available on the internet. For example, the Clinical Trials Transformation Initiative is providing this brainstorming tool I mentioned before. You have the Medicines and Healthcare Product Regulatory Agencies, samples of risk assessments. You have the Accelerate RBM resources. You even have an electronic version of the RACT and the modified version of the RACT Plus, a free version available. It's also I'm providing you with a link so that you can make use of it. Concerning risk management and control, you can check these different methods and mentioned by the ICH guideline on quality risk management and the Q9, ICH Q9. And on top of this, I'm giving you some references of guidelines you should know and you should consult before you implement your risk-based approach to quality management, and some further guidance documents and articles that you will find freely available on the internet. In the end, I'm providing you some references concerning the ISO on risk management and even concerning medical devices. With this, I am thanking you for your attention, and I wish you great success with implementation of this risk-based approach and the quality by design approach in your clinical trials. Thank you very much.